All right. So yeah, welcome everybody. It's my great pleasure to be with you for the next hour and to discuss with you PSP. To start with, I would love to get to know who is actually on the line. I count 142 participants, which is great. So maybe we can start with the first question, and you may want to let us know what professional background you're coming from. So this is the questions you can just type in, and that would help me a lot in order to talk to you in the way that is most suitable for your experience background.
So already 80% of the attendees have responded, and I will show now the results.
All right, so we have adult neurologists, child neurologists, 6%, residents, some physiotherapists, some speech therapists, 1%, researcher scientists, 4%, patient representative, 2%, and PhD students. All right, so let me start by giving you an outline of what we will discuss now in the next hour. First, I will give you a very high-level overview about what PSP actually is in order to go right into matters. And then we'll take a step back and I'll talk a bit more in general about tauopathies, the disease mechanisms and the broader group of diseases that PSP is falling into. Then we'll talk about how to make a clinical diagnosis of PSP. That is talking you about the difficulties and solutions and challenges and where is the level of clinical expertise that we can obtain by now. Also about biomarkers and imaging biomarkers, fluid biomarkers that we can make use of in order to make a better diagnosis. I have some video illustrations in order to talk to you about the presentation, the spectrum of clinical manifestations, the molecular diagnosis, and then in the very end, I'll go to take a deeper dive with you into the molecular mechanisms, why the neurons are degenerating, and to identify possible molecular targets, how we can try to stop the disease from progressing. And in the very end, I'll give you a brief overview about the clinical trials that are currently ongoing with the aim to stop disease progression. It's a very active field of research, so I think you will appreciate that this is an interesting topic to work about. By the end of the presentation, I would like you to be able to make a clinical diagnosis of PSP in a reliable manner. I would like you to know about the current status of the diagnostic modalities that we can make use of. That is MR imaging, that is tau PET in the research status, that is fluid biomarkers. And I would love to you to know a bit about the molecular pathogenesis and how we can actually try to prevent the disease from progressing, and what from these disease-modifying approaches has already entered into clinical trials. So this is what we want to obtain as knowledge to take away from that lecture. Now, the title of my presentation is PSP, Progressive Supranuclear Palsy, the name of the disease. And I'll give you an update on diagnostics, biomarkers, and therapies. So let's take a small jump back in time, 55 years earlier on. There was a group of Canadian neurologists, John Steele, Clifford Richardson, and Jerry Olszewski. They were observant in their clinical routine, and they observed a phenotype which occurred rarely, but repeatedly. And so they wrote a small case study, which was actually already very detailed, and most of what they describe actually holds still true today. So the clinical phenotype of what they reported was an adult onset progressive neurodegenerative condition, which was sporadic in nature, and which was characterized by an inability of the patients to look up and down with their eyes. They termed that supranuclear ophthalmoplegia because the damage seemed to be higher up than the ocular nerve nuclei. So a convergent ocular motor movement disorder. And that movement disorder actually affected mainly the vertical gaze rather than the horizontal gaze, which is helpful for clinical differential diagnosis. The patients also displayed pseudobulbar palsy leading to dysarthria and dysphagia, and they had an akinetic rigid syndrome, which mainly affected the trunk and the neck of the patients rather than the arms and the legs. They had observed also a degree of dementia in these patients, but they noted that this was not the predominant clinical features. It remained mild in most of the cases. So this clinical description is still what we refer to as Richardson syndrome, according to the name of the second author of the paper. And today it's operationalized by identification of these two main clinical issues. That's first, oculomotor dysfunction, and second, postural instability leading to falls. Oculomotor dysfunction can come with three different distinctions, gradings of severity. The first is a vertical supranuclear gaze palsy, which means an impairment in the range of vertical gaze palsy. The second is slow velocity of vertical saccades, which means that the amplitude is still okay in the full range, but that the saccadic velocity is reduced in speed. That's occurring earlier in the clinical course than the reduction in the amplitude. And then in the very early clinical course, some but not all patients show macro square wave jerks. This is if you ask the patient to look straight into your eyes, and the patient's eyes are not standing still, but they are making little jerky movements to the left and right and come back to the midline. That's what you commonly, but not always, observe in PSP patients in the early clinical course, and might help you to differentiate these diseases from patients with Parkinson's disease, for example. And postural instability comes also in distinct severity grades. Today we ask for spontaneous unprovoked falls that lead to falls in patients without any identifiable obstacle, no loss of consciousness, no hurdle that actually precipitates the falls. It's just unprovoked repeated falls. Earlier than the occurrence of falls, patients usually show on the pull test that you as a doctor examine in the patients, they show a loss of balance, and they would not be able to regain their balance unless you help them actually to stabilize them. And even earlier than that, these patients, the pull test would require more than 2 steps backwards before they regain balance. These early stage patients would regain balance actually, but they would take a couple of extra steps as compared to healthy adult persons. Now, in the initial paper, Steele, Richardson, and Olszewski also looked at the autopsy of these patients. They did not yet have very sophisticated molecular biological methods, but by simple histological methods, they graded the severity of neuronal loss in these patients. And they identified a pattern which we still recognize today with our more sophisticated methods. So here in color codings, the darker, the more nerve cell loss. Caudate putamen more expressed in the pallidum and subthalamic nucleus than in the midbrain. You would have a strong affection of the tectum, lamina quadrigemina, the substantia nigra pars compacta. Then in the pons, you would have an affection of the pontine nuclei and the periaqueductal gray. That's the typical pattern they observed in their paper. And if we use today, for example, MRI volumetry, morphometry to quantify and color code the severity of atrophy, then we see very much the same pattern in the basal ganglia and brainstem. But we are also able to identify a more pronounced cortical atrophy, which is affecting mainly the frontal lobe in these patients. So on the microscopical level, Steele, Richardson, and Olszewski at the time used silver impregnation to identify protein aggregates, and they identified neurofibrillary tangles, exactly as they would be present also in Alzheimer's disease. And now later, over the years, it became evident that these tangles are composed of a protein called tau. It's the same protein that causes Alzheimer's disease. It's hyperphosphorylated as much as it is in Alzheimer's disease. It has a different anatomical distribution within the brain, unlike in Alzheimer's disease. And also, unlike in Alzheimer's disease, it does not only affect neurons, but it also affects astrocytes, as you can see here on top, for example, and it affects oligodendrocytes. These two glial components of PSP are not observed in Alzheimer's and helps to distinguish. Now, what is the tau protein? That's a common and abundant protein which is expressed in the axons of neurons. It has 3 or 4 repeat regions. That's domains which are very similar in the molecular arrangement in the amino acid sequence, and therefore they are called repeat domains. And that's actually the domains by which the tau protein binds to microtubules, to tubulin monomers, to stabilize the microtubules, which is actually the cytoskeleton of the cell, in particular of the neurons in the axon. And then these tau molecules have a projection domain, and we don't know precisely what this projection domain is doing, but apparently it is stabilizing the distance between the microtubule railways in order to keep them straightly aligned. So this is the molecule, and this is the function. And when things go wrong, then these tau molecules can actually aggregate. So this is a schematic representation of 1, 2, 3 tau molecules in a straight view and an axial view. And you see that particularly these repeat domains are actually forming these fibrillar aggregates. This is the type of aggregates, the clumps that you can then see by immune histochemistry in the cells, and that cause neurons from degeneration to degenerate. The tau molecule comes in 6 isoforms, and the splicing takes place on one gene. The gene is called MAPT, microtubule-associated protein tau, which is encoded on chromosome 17. And you can have an N-terminal splicing of 2 exons and a mid-region splicing, which is exon 10, and exon 10 splicing in or out actually determines whether tau has 3 microtubule binding repeats as displayed here, or whether it has 4 microtubule binding repeats. And in total, you then have 3 isoforms with 3 repeats and 3 isoforms with 4 repeats. So that's the tau protein. That's the tau protein isoforms. And it's very surprising, interesting, and still not really fully understood, but it's a classical and repeatable observation that distinct isoforms are aggregating in distinct diseases to a differential degree. So there are diseases like Pick's disease, where mainly the 3 repeat tau isoforms are causing degeneration and are aggregating. There are diseases like Alzheimer's disease, where both 4 repeat and 3 repeat isoforms are aggregating to the same degree. And then there is diseases which are predominantly caused by aggregation of 4 repeat tau. That's called the 4 repeat tauopathies. And the diseases we are discussing today, progressive supranuclear palsy, corticobasal degeneration, are falling into that group of 4 repeat tauopathies. Now, when you use different antibodies to stain either 3 repeat tau or 4 repeat tau, you can get different patterns of aggregates in the brain. And these are being used to classify distinct diseases. The first big and most predominant tauopathy is Alzheimer's disease on top. Alzheimer's is considered to be a secondary tauopathy because we and most people in the field would say it's caused by a primary aggregation of amyloid beta. And then on a secondary downstream event, tau is caused to aggregate. That's why we consider Alzheimer's disease as a secondary mixed 3 and 4 repeat tauopathy. The other tauopathies listed here are considered as primary tauopathies because there is predominantly tau aggregating. And here we have the group of 3 repeat, with Pick's disease being a prominent example. And then we have the 4 repeat tauopathies with PSP, progressive supranuclear palsy, corticobasal degeneration, argyrophilic brain disease, and glial globular tauopathy as predominant examples. You can see from the cytoplasmic aggregation pattern of tau in these cells that neuropathologists use this pattern in order to make the distinction of the diseases. So PSP is defined and classified by neuropathologists as a disease which is almost exclusively displaying tufted astrocytes. So this is tau aggregating in astrocytes in the proximal branches, as seen here. Corticobasal degeneration, in contrast, has tau aggregating in the distal proportions of the astrocytes, and that's then called plaque-like astrocytes. And you have argyrophilic grain disease, where you have these punctae, that's grains, and you have these globuli, which is defining glial globular tauopathy. So this is, again, the summary. We have PSP defined as a 4-repeat tauopathy, a primary tauopathy with neuronal tau aggregates in forms of tangles, pretangles, and neuronal axonal threads. And then we have oligodendrocytic tau in the shape of coiled bodies, and we have astrocytic tau in the shape of tufted astrocytes. So this is the disease, when we observe patients over time, or if we observe patients cross-sectionally at different stages of the disease, it becomes apparent that tau is also spreading, as it does in Alzheimer's disease, and as synuclein spreads in Parkinson's disease. The interesting thing is that tau is spreading in a different pattern according to what cell type we are looking for. So neuronal tau spreads apparently independent of astroglial tau and of oligodendroglial tau. But this spreading and the neuroanatomical distribution of tau lesion defines the distribution of neuronal dysfunction. And this distribution again then defines the clinical symptoms these patients are displaying. So let me see whether you are still all awake. There's 163 participants, so I expect you 163 replies to the next question. The summary, MAPT, the gene encoding the tau gene, how much isoforms does it encode? There is the possibility 1 3R isoform, 3 3R isoforms, 1 4R isoform, 3 4R isoforms, 1 3R and 1 4R isoform, or 3 3R and 3 4R isoforms. What is your thought about that question?
Okay, I will leave the poll open another 15 seconds, and then we can show the results. Okay, great. So here are the results.
All right, so the majority is right. 3 3R and 3 4R isoforms. That's the right, correct answer. Okay, next question. Just jump to the next question. Now, PSP is caused by tau aggregation. What is your take? Is it a secondary mixed 3R and 4R tauopathy? Is it a secondary pure 3R tauopathy? A secondary pure 4R tauopathy? A primary mixed 3R 4R tauopathy? A primary pure 3R tauopathy? Or a primary pure 4R tauopathy? We look forward to get your thoughts on that.
Okay, 15 seconds left. And here are the results.
All right, again, the majority is right. It's a primary pure 4R tauopathy. Okay, so that was an easy exercise. Now, let's resume. So we have defined a clinical syndrome, which we refer to as Richardson syndrome, which is a combination of eye movement problems and postural instability. On the other hand, we have defined a neuropathological definition. That's a disease caused by 4 repeat tau aggregates, which affects neurons, astrocytes, and oligodendrocytes. How do these two relate to one another? Well, in the beginning, a while ago, researchers thought that it's identical, the neuropathological definition and the clinical syndrome. And therefore, the NINDS SPSP research group was defining the diagnostic criteria for clinical diagnosis of PSP by the combination of these two main clinical features: vertical supranuclear palsy, reduction in the amplitude of eye movements, plus postural instability with falls in the first year. Very simple, only two symptoms, and the researcher thought that this predicts the tauopathy in the brain of the patients. Then we had a possible diagnostic criteria set, which was relying either on one symptom, which is vertical gaze palsy just by itself, or a combination of slowing of vertical saccades plus postural instability within the first years. Too good to be true, very simple, only two symptoms to look for. And how does that actually perform in the attempt to predict the neuropathology with regard to the detection of the tauopathy? So we try to address that question by looking at the clinical diagnosis and contrasted that with the autopsy confirmed diagnosis in 100 PSP patients. And we found actually that the NINDS criteria, as simple as they are excellently specific. So with that clinical diagnosis, you can predict the neuropathological findings by more than 80%. So that's great. And the problem with these criteria was actually the low sensitivity. In the first 2 to 3 years after symptom onset of first PSP-related symptom, these criteria were actually only able to identify 20, 40, 50% of the patients in the early clinical course. And we thought this is really not an ideal situation, particularly when we consider that PSP patients on average have a life expectancy of 6 to 7 years. So by halfway through the disease, we clinicians left with the old diagnostic criteria were only able to make a diagnosis of 50%. That was not what we actually were very happy with. So we looked in our clinical cases and tried to identify what was actually the clinical syndrome that the patients with the definite neuropathological definition of PSP were presenting with in the very first time, so in the first 2 years. And we find, to our surprise, that from the definite PSP patients, only 24% had a Richardson syndrome in the very beginning. 7% had only oculomotor dysfunction. 20% had only postural instability without oculomotor function. 20% looked like Parkinson's disease in the beginning. 12% looked like frontotemporal dementia. 7% like corticobasal syndrome, and 13% were actually unclassifiable according to classical diagnostic categories. So when we went back to our cases and when we looked into the literature, we identified that PSP, defined by neuropathology, can present with a broad spectrum, an unexpectedly broad spectrum of clinical manifestations. So we had the classical phenotypes, which is Richardson syndrome, which is a combination of postural instability and oculomotor dysfunction. But we also had postural instability just by itself and oculomotor dysfunction just by itself. With regard to the motor predominant phenotypes, there was PSP with predominant Parkinsonism, looking like Parkinson's disease. There was progressive gait freezing, a rare symptom, but highly predictive for PSP pathology. There was corticobasal syndrome, that's the motor phenotypes. And then we have the cortical or cognitive phenotypes, that was behavioral variant frontotemporal dementia, progressive non-fluent aphasia, apraxia of speech, and very, very rarely there were also reports of primary lateral sclerosis and cerebellar ataxia. So that is an unexpected, incredibly broad clinical manifestation that we were experiencing. And now we had a look at 200 autopsy confirmed patients with PSP. We classified them by the initial clinical syndrome, Richardson syndrome, frontal lobe syndrome, Parkinsonism, postural instability. And we characterized the neuropathological distribution of the lesions in the brain. And not surprisingly, the lesions and their distributions actually follow pretty much the syndrome that we see. Like in patients with a corticobasal syndrome, we have more of a parietal lobe affection, much more than in Richardson syndrome. Or in Parkinsonism, we have much more of a midbrain predominance, less so of a cortical syndrome. So it's the distribution of tau in the brain and the consecutive neuronal degeneration that dictates the clinical phenotype. So this is one example. What we see on MRI pretty much resembles what we see on the tau distribution in the neuropathology. This is Richardson syndrome's patients. This is a patient with a corticobasal syndrome where we have much more of a cortical distribution, and that again is reflected here in the neuropathology. So question, which of the following, we need a question to the audience again. Which of the following, I think, yeah, here it comes. So which of the following is not a typical clinical manifestation, not a clinical phenotype of PSP? Posterior cortical atrophy, an affection that mainly comes from the occipital lobe. Richardson syndrome, eye movement problems and postural instability. Corticobasal syndrome, a combination of basal ganglionic symptoms with cortical lobar syndromes. Progressive gait freezing, and PSP with predominant Parkinsonism. What is not compatible with a diagnosis of PSP?
Okay, so 70% of the attendees already have responded, and I will let it run for 10 seconds and then I will show the results. Okay, so here are the results.
Yeah, that's absolutely right. Posterior cortical atrophy is very typically associated with Alzheimer's disease, but not with PSP. Good. So now we see that the original diagnostic criteria were made to identify Richardson syndrome, and therefore their sensitivity for Richardson syndrome is great, is really nearly 100%. The problem is the identification of the other phenotypes like oculomotor, Parkinsonism, corticobasal syndrome, frontotemporal dementia. They are really not recognized very well with the NINDS PSP criteria. So the old criteria were specific, but not really functional in the early stage and not very sensitive. And therefore, we aimed to make new diagnostic criteria, which did not only focus Richardson, but also aimed to identify variant PSP syndromes, and especially also the early clinical course, which we termed as being suggestive of PSP. So we assembled a large group of researchers, and yeah, we went into the literature, looked at our own cases, met for 2 days, discussed up and down, and we modified the criteria, the old criteria. We did not want to overthrow them completely, but to build upon them because they were highly appreciated in the community, mainly for their high specificity. So we changed them. What we actually did, we left more or less unchanged the basic structure, which relies on a basic identification of sporadic patients with an age of onset older than 40 years, a gradual disease progression. We had some clinical and imaging mandatory exclusion criteria. We updated context-dependent exclusion criteria. And then we took the old criteria. The NINDS criteria relied on 4 features in 2 domains. The domains were oculomotor dysfunction and postural instability, and they came in 2 diagnostic certainty levels. The first was most predictive vertical supranuclear gaze palsy, reduction in amplitude of the eye movement. And the second was earlier disease stage, the slow velocity of vertical saccades without affecting the range. And the same was true for postural instability. Most predictive was spontaneous unprovoked falls within 3 years of the symptom onset. And the second, a bit less predictive, was a tendency to fall on the pool test. And what we did then, after careful thinking and analysis of the evidence, we added 2 different functional domains. The first is akinesia, in order to accomplish also, incorporate also the Parkinsonian phenotypes, and then the cognitive dysfunction in order to also incorporate the cognitive and cortical predominance types. And then finally, we added a third level of evidence, that's signs and symptoms, which are less specific but very sensitive, particularly for the early clinical phase. So that's what we changed, and I'll just go into the individual features so that you see the functionality of these criteria. So eye movements. We have oculomotor dysfunction as the first core feature, and we have labeled them, these features, by numbers. O1 is most predictive, O2 is a bit less predictive, and O3 is more sensitive, but even less predictive or less specific. So O1, most specific, is vertical gaze palsy. What you see in that patient is an attempt to look up that does not work. He looks left. It looks right, which works, is slow, but in the full range. And then again, the patient tries to look up again, and that does not really work. So this is level O1, vertical gaze palsy. Next one is full range of movement, still possible, but the movement is slow. In a normal person, healthy person, you would see just the starting point and the end point of a saccadic eye movement. And here you can see how you can follow the eye all the way up and down, because this is a very slow movement, but it's intended to be not a pursuit movement. It is intended to be a saccadic movement. So that's an example of slow velocity of vertical saccades. And then the next patient shows you these square wave jerks. You see all these small little jerky movements in the eye while he is holding the eye straight, or while he's actually moving the eye, but he's continuously interrupted by these small jerky movements. That's level O3, vertical square wave jerks. Now let's move forward to postural instability. So unprovoked falls, I don't have to show you a video on that, you can imagine. This is a pool test of a patient where you explain how the test functions. You stand behind the patient, you ask him to stand straight and try not to fall. And here just a small little tip with the patient, and she's falling into my arms. That's P2, postural instability, level 2, tendency to fall on the pool test. And then the third level, this is a patient who is being pulled, and he's perfectly capable of standing straight again, regaining balance, but he takes more than the 2 steps that a normal patient at this age range would actually require. He takes more than 2 steps, and therefore it's a level 3. Now, akinesia. So the first A1 is progressive freezing of gait. That does not occur in all patients, but it occurs in some patients. And if it occurs in the early clinical course, not as in late stage Parkinson's disease, but in the early clinical course, then it's also quite predictive of a PSP pathology. So you see a patient who's walking down the stairs, and now as he enters the floor, he's getting the freezing. And to see he's not very severely affected by Parkinsonism. It's a predominant feature in the early clinical course, progressive freezing of gait, more or less in isolation. That's A1, not very frequent, but highly specific. Then A2, that's the typical Parkinsonism you see in PSP. That's a small axial predominant, non-tremulous akinesia rigidity syndrome, which does not show the typical decrescendo in Parkinson's disease. This does not respond to levodopa. So this is the typical PSP kind of akinetic rigid syndrome. And then A3, that's Parkinsonism, just as you would see that also in Parkinson's disease. This is a left-sided tremor dominant Parkinsonism, bent forward, arm is not swinging, responsive to levodopa. You would certainly make a diagnosis of Parkinson's disease in that patient until the time when he would develop features that more resemble to PSP, like eye movement problems. But this is part of the spectrum that we can observe in PSP, so we have to take that into account for our diagnosis. And then the cognitive dysfunction, the cortical features. The first is a speech and language problem. I just go over that. This is a typical primary progressive aphasia syndrome. Then a frontal lobe syndrome. You can see the applause sign, for example, the patient not being capable of stopping to clap when you ask him to. And then limb apraxia, that's a patient with a left-sided apraxia, not able to copy the figure he's perfectly fine to do in the right side. So that's the cognitive features we see. And how about imaging? Well, we have, of course, midbrain atrophy, which is suggestive. And we have postsynaptic striatal degeneration, which is not occurring in Parkinson's disease. And these two findings are quite supportive in order to make the diagnosis. Just be aware that midbrain atrophy, as much as we associated that with PSP, if it occurs, then it's quite specific for PSP. It's not very often observed in MSA, not very often observed in Parkinson's disease, but it's not highly specific, which means, excuse me, it's not highly sensitive. So if you do not see that atrophy, like in these patients here, you can still have a diagnosis of a PSP. So that's the kind of information we collect clinically with these patients. And now we assemble that information in order to make a graded diagnosis of PSP. And we grade that by level of predictive evidence for the tau pathology that underlies these diseases. So the first is definitive, PSP definite. That's of course when histopathology evidence supports the diagnosis of PSP. Then we have a probable level, which is patients where we have a quite good credibility about their clinical diagnosis. These are well suited for therapeutic studies. Then we have possible PSP, which is appropriate for epidemiological study, less specific but more sensitive. And then finally, we have suggestive of PSP, which is intended to be very sensitive, but at the compromise of a reduced specificity. How do we make these diagnoses? It's actually quite simple. We take our 12 symptoms that we have identified so far, and we highlight the combinations. And in an accompanying table, we have a list of combinations of these features, which then enable you to make a diagnosis of possible or probable PSP, again, in order to attempt predicting the underlying tau pathology. So this is the list. Please don't be shocked if you see that list. I don't know that by heart myself. I looked that up in the publication, but there is a lot of knowledge that went into the generation of that list, and I think it's quite functional. So with regards to the features, the clinical features, I would just like to recommend to you that publication that we just brought out in Parkinsonism-related disorders. It's one of the most downloaded papers there. And we have videos for all of these individual features where you can make use of in order to familiarize yourself with these features in order to identify them in the clinical routine. So that's, for example, one of the videos, just to show you a patient from Japan with a frontal lobe disinhibitory syndrome, which is overstuffing the mouth while eating, so typical frontal presentation of PSP. All right. So how do these diagnostic criteria actually perform? I think this is a quite nice publication from James Rowe's group. He has studied a large number of PSP patients and controls, and he finds that these new MDS criteria have quite a good specificity when the probable and possible criteria are being used. They have a dramatically increased sensitivity as compared to the NINDS criteria. And therefore, they are, in our understanding, quite helpful, probably better than the old criteria, high specificity, better sensitivity. And this is the performance of the suggestive of PSP. So here you see a sensitivity of 100%, which means you identify nearly all patients who have PSP, but the specificity is only 50%. That means, in other words, it helps to identify all patients, but within the group of patients you identify, upon clinical long-term follow-up, you will have 50% who actually turn out not to have PSP. And that's, I think, quite a helpful population if you define your patients with these criteria to make longitudinal studies in order to identify cool biomarkers which help you to sort out which patient actually has PSP or not. Now, what we did was we reshaped the old criteria. We opened them up in order to become more sensitive, particularly in the early stage. And I think what we need now is a clinical long-term validation in order to see how they actually do perform in clinical routine. So what is the status of the molecular diagnosis? That was all clinical work. So you will all know that there is tau PET developments going on. All of the tau PET work is focused on Alzheimer's disease. All traces that are out there have been identified or developed to identify Alzheimer's disease. And this is one that we were using in recent time in our Munich cohort, PI 2620. It works perfectly fine to identify cortical deposits of the mixed 3 or 4 or tau in Alzheimer's, but it does actually also work to a certain degree to identify the basal ganglionic tau in PSP and in corticobasal degeneration. So we are on a good way, but they are not yet ready for clinical routine use. They are not approved for clinical use. It's still a research tool which is on a very good track. How about tau, phospho-tau, CS and neurofilament? Neurofilament is a non-specific neurodegeneration marker, but which actually is also up in PSP, so it might help you to differentiate that from Parkinson's disease. And quite surprisingly, phosphorylated tau, which is known to be upregulated in CSF in Alzheimer's disease, is rather downregulated in PSP. And the lower your phospho-tau, the more rapid your progression of PSP. It's not a diagnostic marker, but it's just good to know that it does not behave the same way as we know it from Alzheimer's disease. There is work going on to identify by mass spec markers, patterns of tau fragments. That's also not ready for routine use, but there is work ongoing. And what is also quite promising, not yet ready for clinical use, is RT-Quick. That's an aggregation assay of the seeds of tau in the CSF in order to see whether there are seeds who would actually induce tau to aggregate in a test tube. That's also not yet ready for clinical use, but I think that's quite exciting research that's ongoing. Current therapy, I'll just go very quickly over that. I'm sure you being mostly clinicians know that we have levodopa, amantadine, amitriptyline, zolpidem, CoQ10, botulinum toxin, but the degree of recommendation is very weak. There is very little evidence to support that for clinical use, but you should still give each patient a try. And now the question, what is in the pipeline? So you know that tau is the culprit. Tau is the bad guy, which kills neurons in PSP. So there is a lot of genetic and epigenetic evidence that tau is upregulated in the brains of the patients. We know that from GWAS studies and DNA methylation studies. There is also evidence that tau isoform composition goes wrong. So we have higher predominance to the 4 repeat tau isoforms. There are some post-translational modifications like phosphorylation, which go wrong in PSP. And there is tau degradation deficits. We know that from genetic studies in PSP. And what we also know is that tau is being released from the cells, taken up into the neighboring cells, and then causes the spread of the disease pathogenesis from diseased cells into previously healthy cells. So a lot of research effort goes into the nature of the extracellular tau, e-tau, in order to identify what is actually the molecular identity of the spreading specimen in tau. So question, what is the pathogenesis of 4 repeat tauopathies? Is it genetic variation at the tau gene, MAPT? Is it epigenetic changes at the gene called DLX1? Is it altered tau splicing? Post-translational modifications of tau? Alterations in the protein degradation pathway? Intercellular spreading of tau? Sorry, that's written wrong. Intercellular. Yeah, no, it's all right. So it's between the cells. Or is it all of these mechanisms which contribute to the pathogenesis of tauopathies?
Okay, 10 seconds left. So 70% of the attendees have responded. Here are the results.
Yeah, that's right. Perfect. So it's really a mix of everything. It's genetic, epigenetic, post-translational modification, degradation, and spreading. All right, so very much attendance here. Next question, next question. What is the clinical phenotype actually of tauopathies, primarily driven by? So what causes whether a patient has Parkinsonism or whether a patient has a frontal lobe deficit or Richardson syndrome? Is it genetic variants at the tau locus? Is it more a genetic determination? The histopathological hallmark, so is it more the microglia or is it more the astrocytes or neurons? Or is it the topography of the cerebral aggregation? So which region of the brain is mainly affected by tau pathology? Or is it the age of onset? What do you think?
Okay, 10 seconds left. And here are the results.
Yeah, that's absolutely right. Like in nearly all neurodegenerative diseases, where within the brain you have the damage that dictates the symptom. Yeah, a very simple and general rule. All right, so then in the last couple of slides, I would just like to show you, we have learned about the symptoms, we have learned about the molecular pathogenesis, and now I'll discuss briefly where we are with development of therapies. So this is a result of a antibody study, a tau targeting antibody, which targets the N-terminus of human tau, which is just in press in the Lancet Neurology. Where we have studied a phase 2 randomized placebo-controlled multicenter trial in 2 different doses of that antibody compared to placebo. We included Richardson syndrome patients in the early clinical stage, and the primary outcome was the PSP rating scale. Here we have to say that unfortunately there was not a sign of clinical efficacy of that antibody. We had good tolerance, but there was no meaningful benefit that these patients experienced. So we do not think that this is the end of antibody treatments. This antibody study was testing the hypothesis whether a blockage of tau transmission from one cell to the neighboring cells by extracting extracellular e-tau with antibodies from the brain would actually be able to stop. Now, what was wrong in that trial? Well, nothing was really wrong, but the question is, what could we do better? First, I think we could go for earlier stage patients. We could go for longer disease duration. We only had a catchment of 60% of the extracellular tau in CSF, which might not have been sufficient. And maybe and foremost, the epitope might have been wrong. So this is a new antibody that we just have in the phase 1 trial, which is a phase 1b trial, a monoclonal antibody. And the antibody I was just alluding to was an N-terminal antibody. And this new antibody targets the mid-region of the brain, just right to the aggregation-prone domain. So it could well be that this antibody is performing better than the former antibody, and it does so actually in cellular assays. So it's blocking the spreading of tau to a much more profound degree. And we are hopeful, optimists that this antibody might perform better. We have that just ongoing in a phase 1 trial. Now, the other approach that is awaiting to enter into clinical trials is an enzyme which blocks glucosylation of tau. So this is the normal tau protein, and you can have the phosphorylation, which then actually leads to an aggregation of tau. And if you block the phosphorylation by having a sugar molecule here, then you can prevent aggregation. And this is an enzymatically controlled process, which can be blocked by inhibitors. So that's developments, which is called OGA inhibitors. Watch out for these trials. I think there is some coming up in the next time. And then you know also that we can try to silence disease-causing proteins. That seems to work also in the context of tauopathies. So antisense oligonucleotides work in mice. And there is some trials that are upcoming to test whether ASOs, antisense oligonucleotides, can also help in PSP. So with that, I'm through, and I'd like to thank you for your attention. Just brief summary. I hope you memorize that PSP is a prototypical primary 4-repeat tauopathy. We will be able to classify now tauopathies as primary and secondary. That's 3R and 4R tauopathies. You have learned about the performance of the NINDS criteria and the MDS criteria to make a clinical diagnosis. You know now about the existence of that article in Parkinsonism related disorders, where you can have free access to tons of videos on PSP to familiarize yourself with the phenotype. You know a bit about molecular diagnostics, which are not ready for clinical prime time, but which are in scientific development. You know about molecular disease mechanisms and how we can make use to develop new therapies with antibodies, antisense oligonucleotides, and OGA inhibitors. Yeah, with that, I like to thank my team, of course, who supports me in my work, and you for your attention. And yeah, that's my talk. I would like to take the opportunity to remind you of the next talk, which is on genetic dystonia and treatment on February 2nd, same channel. So be with us. We look forward to have you also on the seminary. If there is questions from your side, I'm happy to answer these now.
Yes, we have already received several questions. Thank you, Günter, for the very clear and instructive presentation. So the first one, can you go into detail about prodromal symptoms of PSP?
The prodromal symptoms are more or less what I lined here in the third level of our 12 fields table. So it's the symptoms that refer to different functional domains. In the eye movements, we have the square wave jerks. In the postural instability domain, we have mainly the small balance problems that patients experience often as dizziness. If you do the pull test and you have more than 2 steps to the backward, in the context of cognitive, it can be actually quite widespread. Very often patients refer to a impaired verbal fluency and an apathy in the very early clinical course. But maybe you want, we have just a couple of minutes left. Maybe you want to look into that table. It's actually all written there, and look into the video atlas. You have excellent video examples for these early clinical manifestations.
Mm-hmm. Okay, so idiopathic PLS versus PSP PLS. Do you have any neuroimaging clues suggestive of PSP PLS or associated clinical features like VSG, cognitive deficits, etc.?
Yeah, it's an excellent question, an important question, but I have none, because I think there are really no good clinical clues to differentiate these two entities. It's a pathological diagnosis for the time being, until we have good biomarkers.
Mm-hmm. Thanks for your talk. It was of great interest. I would like to ask about serum biomarkers of PSP regarding the differential diagnosis with PD and other atypical Parkinsonisms. Would you consider neurofilament light chain as an interesting and applicable option for clinical routine and/or trials?
Well, I think NFL works quite good in order to make a distinction between PSP and Parkinson's disease, but not so much to differentiate that from the other atypical Parkinson syndromes, because it's also upregulated in CBD and in MSA. So if you want to rule out in a PSP patient, for example, Parkinson's disease, that might be of help, but not with regard to the other rapidly progressive diseases. In these diseases, I personally trust most the clinical experience. I think we all do look forward to these molecular biomarkers, but for the time being, I think the best you can make use of is actually the expertise of a good clinician.
Mm-hmm. Another one, is there evidence of familial aggregation of PSP or an association with pathologies with a genetic component?
Well, there is a nice paper of John van Sweeten's group in the Netherlands, which does which does show that people who have PSP have a somewhat increased risk in general in the family to have additional cases. That's related mainly to the H1 haplotype at the MAPT locus, which is a factor also predisposing for Parkinson's disease, PSP, and also some association to Alzheimer's disease. Nobody really knows why that is the case, but there is a small but yeah, maybe not clinically highly relevant, but statistically significant association of an increased general risk for neurodegenerative diseases. But usually PSP is a sporadic disease, so it's not an inheritable condition.
Mm-hmm. Are there any dietary or lifestyle recommendations for these patients?
Do not eat Annona muricata fruits because they trigger PSP-like disease in Guadeloupe. Apart from that, just stay healthy and keep on moving.
Okay, any hypothesis about why p-tau 181 decreases with disease severity?
Not really. It's a $100 million question. I have no idea. It just tells us that it's an absolutely different molecular mechanism of action as opposed to Alzheimer's disease, but nobody knows.
Mm-hmm. How many patients with PSP AOS have you seen at your center? What was the phenotype development over time? I wonder, as at our center in Danzig, we saw none, only PSP PNFA. I think 2 out of about 40 PSP cases, and PSP CBS, and no pure PSP AOS.
Yeah, so AOS is apraxia of speech. That's indeed quite rare to see. I personally have seen only 1 or 2 patients in my lifetime, I have to confess. I see quite a number of patients with progressive aphasia, non-fluent aphasia that's can be observed. And also in Richardson syndrome, the non-fluent aphasia is a concomitant clinical feature, but it is convincingly demonstrated by very good and experienced clinicians, foremost Keith Joseph from the Mayo Clinic Jacksonville, that AOS, apraxia of speech, is associated with PSP pathology. It's rare, but it occurs. But I have actually seen only very few patients myself. I agree with your observation on the frequency of these phenotypes.
Mm-hmm. Okay, thank you. I don't know how much time you have left, Günter, but I would take another 2 questions. I mean, there are many of them still.
Go ahead. Yeah.
Yes. Do you think it is possible that the non-RS subtypes could be people earlier in the disease course decompensating for some reason earlier in terms of clinical symptomatology?
Well, I told you already, my understanding is that the spreading of tau in the brain is taking a different path because it would start in different points and probably follow different tracks of axonal connectivity. There are 2 competing hypotheses. The first is what you have just raised, that there is different degrees of vulnerability and resilience, resistance in the brain of these patients. I have more the tendency to believe, but I have no proof for that, that the spreading pattern of tau according to axonal pathways, depending on where by chance the pathology starts in the brain, is actually deciding what phenotype these patients have.
Mm-hmm. In terms of clinical trial design, is there a difficulty in identifying early PSP patients who may receive the largest benefit from a potential therapy? Additionally, what length of a trial, weeks or years, might be required to see a benefit on the rating scale?
Yeah, the first question, yes, I think there is a problem with regards to the identification of patients very early on. The problem is, I think, the referral patterns. These patients usually see the general practitioners and go to neurologists. I think the issue is not so much that they could not be identified. They usually have the clinical symptoms. It just requires actually clinical experts like you, like us, to actually identify these features. So I think the key to identify patients earlier is a good clinical training for experts like you, like through the ERN-RND network, in order to make the diagnosis early on. So the patients seek for help and we need to provide that to them. That's the key, how to identify these patients earlier for clinical trials. The second part, how long needs the trial be? I mean, we have done power calculations. In general, if you start taking a drug treatment, you treat the patient for 12 months and the drug starts to function by 30% from the very first day, then 12 months observation period would be sufficient. However, we do not know how long a drug actually takes until it turns out to be efficacious, and therefore newer trial designs also consider longer observation periods, such as 1.5 or 2 years. But that's always a trade-off because patients deteriorate very severely, sometimes after 12, 18, 24 months. And so you might lose them from follow-up, or you might have problems with ceiling effects of your scales. So I think a trade-off would be 18 months, which is quite good. 12 months might be too short, but well, that's the range that's currently commonly used in clinical trials.
Mm-hmm. With regard to LG-LON5 tauopathy, what is the role of inflammatory changes in PSP?
Difficult question. So the IG-LON5 refers to a rare condition which has IG-LON5 antibodies with sleep disturbances and very often also clinical symptoms overlapping with the PSP phenotype. And since this is considered to be a primary immune-mediated mechanism that causes a secondary tauopathy, there is the hypothesis that immune mechanisms can drive tauopathies. I think in PSP, which has overlapping clinical features and overlapping neuropathology, the genetic evidence mainly is so strong that the strongest driver for PSP pathophysiology, in my understanding, is tau expression and not primarily immune-mediated mechanisms. That just tells us that tau pathology can be triggered by different pathways, amyloid beta in Alzheimer's disease, inflammation in the IG-LON5 tauopathy, and I think primary tauopathy in PSP.
Mm-hmm. Are there non-motor symptoms for PSP?
Yeah, I think I refer you to the table again. There is the entire column of cognitive features, for example, which are non-motor.
Yeah, and just for like a reminder for everybody, we are recording this webinar and it will be published on our website for all those who could not attend or could not see the whole webinar. So another question, any information on SV2A PET imaging on PSP progression? Could it be useful for early clinical trials?
Well, there is some publications actually. It's axonal connectivity mainly. I think it's early times. There is only very limited information. I think it's interesting to follow, but I think I would not feel what is missing in my understanding is longitudinal data, but they are being generated. So I think it's an interesting, attractive candidate, but not for the moment, but maybe for the near future.
Yes, okay. And another one through the chat. Any update you can give on AZP 2006 trials in France? Also, are there current trials happening in India for this?
Well, the AltProtect compound, there is some information in clinicaltrials.gov. There is not, I'm on the advisory board of the company, so I cannot tell more than that, but the publicly available information on the compound is displayed on clinicaltrials.gov. And about India, I am not aware. I'm sorry, but I'm not aware of any studies that would be ongoing there.
Okay, so thank you very much. We don't have any further questions. You have answered a record of 15 questions, so this was great. A lot of interest by our attendees for this topic. So thank you again, everybody. Thank you, Günter, and stay tuned. We see each other next week for the next webinar.
Yeah.
Bye-bye.
Thank you. Bye-bye.